Let's learn about mRNA vaccines and how they work. Vaccines are a familiar concept for many of us, whether we've received them during our high school years or due to the recent COVID-19 pandemic. But have you ever wondered, what exactly are vaccines and how do they work? Generally, vaccinations work by injecting a harmless antigen associated with a specific pathogen to stimulate the immune response to produce multiple memory B and T cells. An antigen is any particle foreign to your body's internal environment, such as a protein found on a virus. The production of memory cells result in a more efficient immune response during exposure to the actual pathogen during infection. During protein synthesis, mRNA is produced in the nucleus for transcription of the DNA and then transported out of the nucleus. Then it binds to a ribosome and is translated into a polypeptide chain through the assistance of tRNA. The polypeptide chain then folds depending on the amino acid's properties, synthesizing a protein. The COVID-19 vaccine stems from this biological process, and pharmaceutical companies including Moderna and Fayza biotech have produced vaccines utilizing mRNA copies from the coronavirus coding for spike proteins to elicit an immune response. You see, as much as we all dreaded COVID-19, it actually stimulated deeper research into this amazing technology. So let's briefly go over the development of mRNA vaccines. Much can be learned from its development history, as it wasn't until the COVID-19 outbreak where widespread use of this technology was implemented into the treatment of COVID-19 in the general population. Contrary to what many may think, mRNA is not a recent discovery. The developments of the COVID-19 vaccines by Moderna and Fayza biotech were built upon almost 3 decades worth of pre-existing research and knowledge relating to mRNA. One of the earliest clinical trials relating to the mRNA and its use in vaccines could be traced all the way back to an investigation in the 1990s. Mice were injected with mRNA and DNA, and the results recorded showed that the genetic material that was injected was able to be translated into a protein structure, much like how our modern mRNA vaccines function. This investigation also revealed some early limitations of mRNA, notably its immunogenicity and instability. Once the protein had been synthesized by the mRNA, the mice's immune system would be alerted and start to create antibodies for the protein. This could result in a range of side effects with differing severity in humans. Further research and developments have allowed researchers to find ways around the dilemma. How effective are mRNA vaccines? mRNA vaccines are found to have a high efficacy rate. In a study conducted by Pollack, examining the effectiveness and safety of the BNT162b2 mRNA vaccine in combating COVID-19, the vaccine was found to be approximately 95% effective in preventing COVID infections. One reason for this heavily attributed to the lipid nanoparticles that surround the mRNA, which increases cell delivery efficiency and prevents it from being rapidly degraded. What are some benefits of the mRNA vaccines? mRNA vaccines are safer since they do not contain the actual pathogen, but only a segment of its genetic code. This means that they are harmless and have minimal risk of causing infection. Unlike traditional vaccines that require the use of animal cell cultures, which means mRNA vaccines are significantly less time-consuming and simpler to produce. In the lab, the DNA that the mRNA is made from can be synthesized and be sent by a computer across the globe in an instant. Multiple clinical trials are evidence of the high rate of success of mRNA vaccines. One such example is the study conducted by Pollack 2020, which examined the effectiveness and safety of the BNT162b2 mRNA vaccine in combating COVID-19 infection through a randomized double-blind study involving participants equal to or over the age of 16. The occurrence of confirmed COVID-19 instances at least 7 days after the second treatment was the main objective. The secondary goals were the vaccine safety and severe COVID-19 infections in people who had previously contracted SARS-CoV-2, median of 2 months. According to the study, the BNT162b2 mRNA vaccine had a 95% efficacy after the second dosage in preventing COVID-19 infection. Moreover, no severe COVID-19 instances were reported in the vaccine group compared to 8 cases in the placebo group, demonstrating the vaccine's effectiveness in preventing these cases. Additionally, among those who had previously contracted the virus, the vaccine successfully averted COVID-19 infections. The study also revealed no documented significant side effects, and the vaccination was typically well tolerated. However, further research is required to understand the safety and efficacy in groups not included in this trial, such as pregnant women or children, and for periods over 2 months. Nevertheless, this study provides important insights into the safety and efficacy of the BNT162b2 mRNA vaccine in preventing COVID-19 infection, which has important implications for developing and distributing vaccines in a global pandemic. Limitation. However, despite the many benefits, there are still some limitations to consider with the use of mRNA vaccines that scientists and pharmaceutical companies are working on minimizing. Firstly, mRNA is very fragile. This property can be traced back to the structural differences that exist between RNA and DNA. The deoxyribose sugar in DNA contains a hydrogen atom on carbon 2 of the pentose ring, whereas the ribose sugar in RNA contains a hydroxyl group on the same position. The presence of this hydroxide makes RNA more likely to undergo hydrolysis, the breaking of a covalent by the addition of a water molecule. Furthermore, there are enzymes in the environment that break down the mRNA. Enzymes are macromolecules that speed up chemical reactions and have a narrow range of temperatures in which they're able to function. As a result, mRNA vaccines must be kept at extreme temperatures to slow down all chemical reactions and prevent the breakdown of the mRNA molecule. For example, Pfizer-BioNTech's mRNA vaccine requires refrigeration at -70 degrees Celsius. In normal refrigerators, these vaccines would indefinitely degrade after 5 days. This raised concerns regarding shipping and maintenance. Fortunately, the pharmaceutical company Moderna claims their vaccine can be maintained at most home or medical freezer temperatures for up to 6 months for shipping and long-term storage. Simultaneously, Pfizer-BioNTech has been working on developing dry ice shipping containers for the vaccine. Another limitation of mRNA vaccines is addressed in the article Antibodies and T-cell immune responses following mRNA COVID-19 vaccinations in patients with cancer, written by Sidis E. and others. As the name suggests, a clinical trial was conducted on the efficacy and durability of mRNA vaccines for hematologic cancer patients, specifically through both humoral and cell-mediated response assessment, and the findings suggest the vaccine to be effective to a minor extent. The results demonstrated an inability to elicit an immune response in some of the patients who received chemotherapeutic or targeted agents. The reason for this is yet unknown and hence requires additional research to cater for cancer patients. The results for durability of the immune response in cancer patients receiving the vaccine were also lower than expected, indicating the need for further research on the cause. Alignment with BABS themes. Our topic aligns with several BABS1201 themes, including macromolecules and polypeptide synthesis. mRNA is a form of RNA which makes it a nucleic acid and hence a type of macromolecule. mRNA is produced via transcription in the nucleus, and then mRNA undergoes translation to produce a polypeptide chain of amino acids. This polypeptide chain folds depending on its amino acid R groups into a specific protein, which is another type of macromolecule. The main role of mRNA vaccines is to synthesize antigenic proteins in another organism to elicit an immune response via polypeptide synthesis and protein folding. Gene expression and cell integrity are also relevant BABS1201 topics to mRNA vaccine. Specifically, the protein produced has specific structural and functional features which are heavily reliant on the mRNA-based sequence due to the amino acid R group properties as we discussed previously. Hence, the gene coded for the mRNA strand is expressed. Additionally, the BABS cell integrity lecture is essential for us to realize that since the cell membrane is semi-permeable and majorly allows the passive transport of small non-polar molecules, mRNA cannot pass through the membrane without assistance, and hence a lipid nanoparticle is used to perform endotosis of the target mRNA strand into the cell. And that's all from us. We covered the development, function, and efficacy of mRNA vaccines, including the pros and cons. We hope you enjoyed and learned something new today.